A very warm good afternoon to all of you. So I'll just directly start into my work since I have like time limit. So I'm Nikita. I'm a research fellow in LKC Medicine, NTU, Singapore. And today I'm going to give a brief introduction into the work which we're carrying out for missense variants using the deep mutational scanning data. So starting with it, like as we have already discussed a lot about all the sequencing studies and all. So now we know that we get to know, or from all these different sequencing studies, we are getting a lot of variants, especially the missense variants. And one of the major challenge here is identifying the pathogenicity of those variants. So this is mainly because we cannot, go and do the experimental testing of each and every variant, and hence a lot of variants are being classified as a variant of uncertain significance. And that is why we need some computational tools to predict the effect of these variants and to show if there are any impact of these variants in the protein. So we collectively call this computational tools as variant effect predictors, which takes into account information at different levels of the protein, such as the protein sequence information, structure, and all the evolutionary features, and then collectively it helps us in showing if the variant is pathogenic or not. But however, there are certain challenges because we don't know the molecular effect or cellular effect of the variants. And also, the VEPs are more effective in predicting the loss of function variants rather than the gain of function variants. So one advancement in this thing, in the variant effect prediction, is the deep mutational scanning, which is basically a high throughput wet lab experiment which generates thousands of all possible variants for the protein, and then it checks the fitness of all the variants. And as a result, we collectively get a data set of variants with the fitness scores. So we get to know like which variant is having, or showing a decreasing effect in the cell, or it is like increasing the effect of the cell. So this is what our main focus of the research is. And we are like collectively working with Prof. Joe Marsh from University of Edinburgh and Prof. Joanne. So what our aim is, we are trying to use all the available DMS data sets for the proteins, and then we are trying to classify the variants, like trying to classify the pathogenic variants based on already known pathogenic variants from ClinVar and the population databases. So we have the access to the SG10K health data set, and along with that, we're also considering 3 other population databases, such as the Mexico City data set and the NOMAD. And we are trying to find out if there are any clinically relevant pathogenic variants in that. And then we are grouping those grouping those variants into the pathogenic groups and the benign group, and then going to the structural properties of those groups. And of course, since time is limited, this is only a part of the structural things which we are doing now, because we have seen that after we have classified the variants into the pathogenic groups and the variants having the immediate effect and also the benign group, we can see that this group of variants are showing a very destabilizing energy compared to the benign group. And of course, we can see a large amount of variants from the pathogenic groups. They are located in the interior regions or the interface region of the protein. So that is one of the more important points, because even sometimes some variant effect predictors, they miss these points because a lot of variants, even if they are present in the interface, they might play a very important role because they can lead to the destruction of those interactions. So this is what we saw from our results. And yeah, of course, on the first plot, you can see the proportion or the distributions of the missense variants in all these different groups. So yeah, it's still ongoing, and we have done a few things, but yeah, hopefully like when the paper will be out, you'll all be able to see the full results of it. So that's all. These are like my lab members, and yeah, the UOE LKC team for the funding and the support, and the SG10K health team, and the NSCC Singapore for providing the supercomputing facilities for carrying out the analysis. So thanks.